He was a summer intern at the Research Experience for Undergraduates internship at the Westport Center in Albany, New York. Where he studied focusing both in data interpretation and prayer in the Arcata, and later spent a semester studying at Nihon University in Mishima, Japan. The experience has played a major role in him earning the prestigious Fulbright Scholarship and the Stuttgart Undergraduate Research Scholarship award. In the fall, Sam will be attending grad school to earn a PhD in biochemistry at the University of Wisconsin-Madison. Please welcome Sam.
Thank you everyone for coming. Today I'm very honored and delighted to give you a talk on a project that I've been working on in the Nicolau lab under Marna Nelson for the past 3 years now, ever since freshman year. We've been focusing on hydrocarbon biosynthesis on the surfaces of maize silks, and I don't have time to tell you everything that we've done in the lab since I've started, but I do have enough time to talk about two major impacts on hydrocarbon biosynthesis, namely the developmental effects and some of the environmental treatments that we have. So first of all, I need to tell you what exactly are these hydrocarbons that we're looking at. So we're looking at these straight chain linear alkanes and alkenes. These are molecules that are basically the simplest organic molecules we can make. There's nothing but carbon and hydrogen atoms. We analyze these molecules through a technique known as gas chromatography mass spectrometry. So we collect the hydrocarbons, we run them through a long column that separates out all of these hydrocarbons based on their physical properties. And then we can take any one of these peaks as they come off the column and fragment them, and the fragmentation pattern tells us what molecule they are. So for example, if we look at nonacosane, or C29 as we call it, we observe this very easy to identify, very signature mass fragmentation pattern. So hydrocarbons are actually, believe it or not, found all over in nature in very discrete tissues. So Micrococcus luteus, Shibonella ododesis, these are two bacteria. You got plants, algae, insects, the eared grebe, and mice, and most other vertebrates. And what's interesting is that these hydrocarbons are found everywhere, but they're only found in discrete places, even within an organism. So, for example, alkanes are found in the myelin sheath of nerves, and like the eared grebe also secretes these hydrocarbons as kind of a coating for the feathers. Even though we've known about the existence of hydrocarbons in biology for almost 30, 40 years now, the way that these things are made is very poorly understood. And so what we do know is that they all come from fatty acid precursors. And for those of you who've taken BBMB 405, you all know that most fatty acids are even numbered because they're synthesized in units of 2 acetyl-CoAs. And so there are 4 proposed methods for hydrocarbon biosynthesis. You could start with 2 fatty acids and put them head to head and get rid of a carbon dioxide and reduce that to an alcohol, get rid of that through dehydration, and end up with an odd number hydrocarbon. The second pathway, you can take just 1 fatty acid and directly take off this carboxylic group, and that gives you an odd number hydrocarbon. The third way is you can reduce the carboxylic group to a carbonyl group and get rid of this part, which gives off carbon monoxide and an odd number hydrocarbon. Finally, you can take this, reduce it to an alcohol, dehydrate it, so instead of giving off carbon dioxide or carbon monoxide, you get rid of water, and this gives you an even number of hydrocarbons, with the same number of hydrocarbons as when you started out. What I think is really weird is that while hydrocarbons are found all over, and they're low abundant molecules, but they're common across biology, we found evidence for pathways in each of these. We found evidence for these different pathways in all different domains of life. And so the ones in bold are organisms where these pathways have been extensively characterized, and then the ones not in bold, there's just molecular evidence. So they do radiolabeling, and they observe carbon monoxide coming off, for example. So with all of those model organisms, why are we using maize cells? maize are really useful to study hydrocarbon biosynthesis, mostly because their cuticle layer contains almost nothing but hydrocarbons. So other types of tissue in the maize contains all of these very hydrophobic molecules in varying abundances. But hydrocarbons are kind of rare, and in the silks we can gather a whole bunch of these silks really easy, and when we do extraction, we can obtain almost pure hydrocarbon. Maize is also a really useful model for the functions of these hydrocarbons, because we don't necessarily know why it's making hydrocarbons in the first place. What we do know is that since they're found on the epicuticular waxes, they probably serve to protect the silk against a variety of abiotic stresses such as solar radiation, desiccation, and biotic stresses such as insects. What I think is neat is that the maize silk is a kind of unique organ. So each of these silks is a conduit for pollen that goes from the tip of the silk all the way down, and it stems from a kernel. And so what it might be is that since these silks have a lot of surface area, they're really prone to drying out and desiccating. They might need a better covering that is better able to protect them from all of these stresses than all of these other fatty acid metabolites. One of the things we know is that genetics plays a very strong role in hydrocarbon biosynthesis. And so the two lines that we work with are B73 and Missouri 17. These are just kind of historically important lines. What people like in Herrera have found before me is that there's very strong genetic input. So B73 typically makes somewhere in the order of 3 to 5 times more hydrocarbons than Missouri 17 or other inbred. When we cross these and make hybrids of the two, what we usually see is that these two hybrids are somewhere intermediate between B73 and Missouri 17. And yeah, so the other thing that I want you to note from this graph is that we've got two sections of maize silk. So we chopped up the silks in two. There are the emerged sections, which are right here. These are all the silks that have grown out into this environment. It's very hot, it's very dry. And then there are these other sections that are still inside the ear that haven't emerged yet. And so these sections are still attached to well, they're obviously attached to the kernel, but they're in this environment that's protected by the husk leaf. And you can see that there might be some effect between emerged and encased silks, where the silks that are encased are not producing very many hydrocarbons at all. This could be due to two major factors. So it could be that it's an environmental thing, where they're coming out into this very different environment. It could also be just the fact that these silks at the top are older than the silks that are still dividing at the ovules in the kernel. And so the next few experiments that I'm going to show you are trying to dissect the silks into more and more gradients, and just to see whether or not both of these factors have an impact and what the relative impacts might be. So we started with two sections of silks. What if we chopped it up into 8? And so in this experiment, you can see we just cut sections perpendicular from the point of emergence, 1, 2, 3, and 4, of both the emerged and the encased. And then we plotted them out after extracting the hydrocarbons and quantifying them. And what we see is if you look at the encased sections, there's a slight increase of hydrocarbons going from the very base of the ear to the tip. But then once you get to the emerged, you see a similar increase, but the amount that it's increasing is just so much larger. So now we know that there's a developmental impact and a strong environmental impact. But if you were to take this ear of corn, completely dehusk it, and lay out the silks, you would see something like this. And what I want you to get from this figure is that we're sampling across two types of developmental gradients. And so the first gradient we have is down the length of the silk. So the silks are youngest in this area, and then as they're growing down, they get older. And then we're also sampling all across the ovule positions. And so at this point, we don't know whether or not the position of the ovule on the ear has any effect and any impact on hydrocarbon accumulation. And so it could be that the silks down here have very little. It could be that these silks up here have a lot, and it's just kind of averaging out. We don't really know. But if we were to look at, say, 20 sections, where we chop up the ear into thirds, and then look down the gradients in each of these sections, we can begin to understand these two developmental impacts on hydrocarbon accumulation. So the next experiment is designed to test this. We're also introducing a third factor, the effect of environment, light versus dark. And so we're using our we're using our 4 genotypes, B73, Missouri 17, and our 2 reciprocal hybrids as for our genetic component. For our developmental component, we will divide the silks up into these 2 different developmental gradients and test whether or not those have an impact. And finally, for environment, we're going to use light versus dark, the illumination state of the years. And so for the light treatment, we cover the silks in a translucent chute bag where light is still passing through. In the dark treatment, we cover the silks in a chute bag, and then we also cover it in these 2 pollination bags. And so there's going to be very little light, if any at all, that's going to reach the silk. What we think is that light might be a trigger where as soon as the silks emerge, they notice the light, and then that serves as a trigger that induces hydrocarbon biosynthesis. So once again, here's our dissection strategy. The first thing we do is we cut the ear into thirds, then we divide the emergent versus encased silks from one another, and then each of these lines to cut the different sections is based on the relative intersections between the ear gradient and the emergence gradient. And so what do we find? We find that the hydrocarbons accumulate down the length of the silk. And so looking only within one section of the ear, we go from A to H. We see that even in the emerged sections, there's still a developmental gradient. For Missouri 17, we actually don't see any effect, statistically speaking. So these, all of these sections are almost uniform to one another. But for B73 and one of the hybrids, we see a very strong effect both in the the encased tissue and the emerged tissue, with the point of emergence being a major factor in determining how strong this effect is. We can also look at the composition. So I mentioned before that these are simple alkanes and alkenes. And so we can look at all of these different classes of hydrocarbons. And so what we see is that from the encased silks, we can look at the relative abundance of the alkene, and we see that there's a slight increase in the emerged tissue. And then once we get to the encased tissue, once we get to the emerged tissue, there's a much stronger effect. So it's increasing by 14% from the from the point of emergence to the very tips of the silk bond. Conversely, when we look at the odd-numbered alkanes, these are the hydrocarbons with no double bond, we see a similar decrease in both of these. And that might be able to tell us something about how these hydrocarbons are biosynthesized and might help us make a metabolic map of from fatty acid to hydrocarbon. When we look at the acropoidal developmental gradient, we see that we can split it up. And so, these 3 sections, there's no silk developmental gradient. We're just looking at the ovule position on the grid. And what we see there is there's a slight environmental effect. So in B73, there was a statistically significant difference from the first section over here, the base of the ear, to the tip of the ear. And while we don't see that in any of the other genotypes, there's a similar pattern from the first to the third, where there's a slight increase, like maybe 20% for this very low one. So the other thing we want to look at is the environmental treatments. And we were actually kind of surprised because we didn't see any major difference in the light and dark treatments. So the first thing we did is we looked at the total abundances of all of these and looked at for any statistical differences. We didn't see anything. Then we split it up into 4 main classes. We see the odd alkenes, the even alkenes, the odd alkanes, the even alkanes. And so we also split these up. We also didn't find anything. There's one little difference down here. We're not sure what to make of it, but the main thing is we don't see very much of a difference at all. The other thing I'd like to point out is that you see most of these is just red and purple. You can barely see any green. I don't even know if you can see the green in there. And then there's a small amount of blue. And so that leads us to believe that there's one of the carbons is being removed as we're making these hydrocarbons. And so it might be analogous to the Arabidopsis pathway, where there's a decarbonylation pathway. I was lucky just a few weeks ago. We finished an experiment this year looking at hydrocarbons, and we found various groups that we could compare from 2012, 2011, and 2010 that all have the same treatments. The only difference was the year-to-year, the field conditions year-to-year. And so one of the things I've been looking at is the effect of rainfall and drought on hydrocarbon accumulation. And what we see is when I look at the rainfall from planting date to the harvest date, there's a very strong effect from in the amount of rainfall. There's a strong correlation between the year-to-year differences in terms of the amount of drought. And so this is consistent with the hypothesis that these hydrocarbons are being accumulated on the silts to protect against drought and all of these other environmental stresses. I'm going to note that this is strictly a correlational study. We can't say anything for certain from here. But what this does tell us is we can just take this data and design growth chamber experiments in order to test these, the effect of rain and the effect of water on hydrocarbon accumulation. So that's cool. There's a bunch of things we can use maize to look for in this environmental system that I don't have time to tell you about. But really quickly, there's about 1 million different environmental effects that might have an impact on hydrocarbon accumulation. So pollination is one I'm looking at right now. It could also be a temperature thing between the emerging case silks. It could be humidity. Insect predation, the insects are more likely to attack the emerged silks because they're not protected by the husk. There's all sorts of factors, and including drought. And so in the coming years, Team Silky is going to be looking at some of these environmental effects and seeing what they do. The same year I did the dissection study, we also looked at a large quantitative trait mapping population where we can look at molecular markers on the B73 and the Missouri 17 genomes and correlate those with increased or decreased hydrocarbon levels. We found some really cool stuff. There's a region of the genome that contains a couple dozen genes, I believe, and it impacts all of the odd-numbered hydrocarbons. So when that gene is present, there's a huge increase in these hydrocarbons. So we found like a really cool master regulator. The fundamental goal of this is to elucidate a metabolic map of hydrocarbon biosynthesis. And so there's a graduate student in our lab who's looking at not just the hydrocarbons, but the other metabolites that maize silk contains. And hopefully using that, we'd be able to go from the fatty acid to, through the intermediates to the eventual hydrocarbon. That would be really cool. In the distant future, we might want to make crops with varying amounts of hydrocarbons in their silks. Because if our hypothesis is true, and these hydrocarbons are there to protect against stresses, especially with drought, like last year was terrible, we might be able to design crops that are better able to resist drought, or predation. All of these things might improve yields together. The ultimate goal, potentially, is biorenewable petroleum. As I said, we find alkanes and alkenes all over petroleum. That's what our gasoline is made of. This one's going to take a lot of work. So we would have to take the genes from maize, improve them. We're only getting micromolar quantities of these hydrocarbons right now. And so we'd improve them, insert them into something, an appropriate vector like yeast or algae, and then maybe one day, if the economics work out, we could get biorenewable petroleum. Still, the potential is there, and if we essentially turn a non-renewable resource into a renewable resource, that would be really cool. So finally, I'd like to make a lot of acknowledgments. Mara Nelson has been a great help ever since I started in the lab. She's taught me so much about being a better scientist, a better writer especially. Nick Lauder is a close collaborator of ours. He helps us a lot with managing the large data sets that we use and a lot of the agronomy stuff too that especially I'm not very familiar with. Alina, Miriam, and Leighton did a lot of field work, and they were involved in dissecting the silks, as we saw earlier, while I was away in Albany. In the Nicolau lab, obviously, I'd like to thank Basil Nicolau for letting me pursue research ever since freshman year and just giving me all sorts of support. When Min was the graduate student, I was first working under, and she taught me a lot about how to extract these hydrocarbons. Finally, I'd like to thank everyone at the Metabolonics Research Laboratory, especially Ann Carrera and Thomas Song, for helping me get through these. I think I've extracted and processed about 2,000 samples right now, and I've ruined a column or two, and they've just been a great help in making sure everything runs smoothly and I get the data that I need. And Song has been great in providing us macros and ways to analyze the data really quickly. So, yeah. With that, thank you very much, and thank you for giving me So that what I was showing you right there was just looking at the emerged portion of the silk, and this was something I threw together, and I think it was last week. So one of the things we want to do is look at the encased silk as well and all the different classes, and then we also want to look at when the rainfall might have an impact. So I can also look at it from like the whole year, ever since like the year before it was planted, because groundwater is important. You could look at it from just like maybe in July or right when the silks emerge, maybe that's the same. There's a lot of ways you can throw it together, but that's one of the reasons why we're excited to start these growth chamber experiments, because then nobody's really tested the impact of drought on these hydrocarbons. And so if we can do that,
So you mentioned your quantitative rate analysis had potential candidate for the master regulator. So what kind of molecule were you finding?
oh, the master regulator?
Yeah.
So yeah, there's a region where there's, I think it's like 22, we have 22 candidate genes. We've also used transcriptomics to narrow it down. And so we looked at between the emerging and the encased, and those 22 genes were both in that region of the genome, and they were expressed in the emerged tissue. And so there's actually a whole bunch of different mutants that you can just buy. And so next year, I think Marna wants to take those and characterize them and see what they might be. It could be, I don't think it's like an actual enzyme that's doing the decarbonylation, because we haven't seen any samples that have no hydrocarbons. And so it's probably just a regulatory work. That's what I think. We don't know. A lot of biotechnology.